Welcome to a presentation from Bio-Rad Laboratories demonstrating the use of the iTaq Universal SYBR One-Step Kit and PrimePCR assays for in vitro drug metabolism enzyme expression analysis. From this presentation, we hope to share with you the benefits of using a quality one-step RT-qPCR kit in conjunction with cell lysates and pre-validated assays. When used together, the simplified qPCR workflow will reduce your time to results while providing data comparable to traditional qPCR methods that involve RNA purification and separate cDNA synthesis. Drug metabolism is regulated at the transcriptional level by nuclear receptors that recognize broad sets of exogenous and endogenous ligands. Studying the expression of genes regulated by these receptors can yield information about the route of drug metabolism and about potential drug-to-drug interactions. Molecular signatures of the expression of drug metabolism enzymes can also be used to investigate potential toxicity of a candidate drug. Drug metabolism enzyme expression profiling for candidate drugs can be performed early in the development using in vitro cellular systems, and because the cost is much lower than that of animal studies or clinical trials, large compound libraries may be screened. Here, we demonstrate the utility of using a one-step RT-qPCR kit to investigate phase 1 drug metabolism enzymes using a set of known inducer compounds. To this end, we screened hepatocyte-like cells for the induction of 7 cytochrome P450 family enzymes in response to a single concentration of 6 compounds. In addition, we evaluated the dose response across a 4,166-fold concentration range. Furthermore, we demonstrate the data concordance between a one-step approach and a two-step approach to emphasize the value of using one-step without compromising data quality. Finally, we demonstrate the use of cell lysates as an alternative to purified RNA, emphasizing the value of removing the purification step to increase throughput and reduce time to results. We chose the one-step RT-qPCR approach to minimize hands-on time and to achieve faster time to results. This is valuable for researchers concerned with the need to quickly screen large sets of samples and targets. The use of a universal RT-qPCR kit allows properly normalized data to be produced on all qPCR instruments, whether or not ROX normalization is required. This enables the use of multiple platforms during larger projects. The use of PrimePCR gene expression assays eliminates the need for tedious assay design and validation. Finally, the use of sample lysates prepared using the iScript sample preparation reagent for RT-qPCR reduces the hands-on time required for sample preparation and makes the entire workflow amenable to liquid handling. The general workflow starts with growing hepatocytes, or hepatocyte-like cells, in 24-well or 96-well cell culture plates. Cells are then incubated with test compounds in induction media for 48 to 72 hours, followed by an appropriate sample preparation method to isolate total RNA for RT-qPCR. To evaluate 2 different sample prep methods, total RNA was purified using Bio-Rad's Aurum Total RNA Mini Kit, while cell lysates were prepared using Bio-Rad's iScript sample preparation reagent for RT-qPCR. One-step RT-qPCR was performed using the iTaq Universal SYBR One-Step Kit, along with PrimePCR SYBR gene expression assays. Data were collected using a CFX 384 real-time PCR instrument and were analyzed using the CFX Manager software. Here is a quick review of the compounds used. Omeprazole, a proton pump inhibitor that is used to treat gastroesophageal reflux disease. It is a known potent inducer of cytochrome P450 family member 1A1 and 1A2, and it is a repressor of cytochrome P450 family member 2C19. Rifampicin, an antibacterial drug used to treat mycobacterium infections such as tuberculosis. It is known to induce a number of phase 1 drug metabolism and drug transport enzymes. Carbamazepine, an anticonvulsant and mood stabilizer that is used to treat seizures and mental conditions. It is a known inducer of cytochrome P450, family member 3A4, and family member 2C19. Artesunate, an anti-malarial drug. Dexamethasone, a synthetic glucocorticoid with potent anti-inflammatory and immunosuppressant properties. Beta-naphthoflavone, an agonist of the aryl hydrocarbon receptor and known inducer of phase 1 drug metabolism enzymes. To reinforce the importance of choosing proper reference genes for expression data normalization, we performed a reference gene selection experiment using the PrimePCR reference genes H384 panel. Hepatocyte-like cells were treated with a single concentration of each of the 6 compounds in induction media. RT-qPCR was performed using 1 nanogram of Orum purified total RNA with the iTaq Universal SYBR 1-step kit. The data were generated on a CFX384 real-time PCR instrument and were analyzed using the GNorm module of Cubase Plus from BioGazelle. 3 reference genes were selected by the experiment based on their low variability across treatments. The panels of drug metabolism and hepatotoxicity assays shown here are available for use with SYBR-based chemistry on Bio-Rad real-time PCR instruments, as well as other platforms. In this experiment, PrimePCR gene expression assays for a set of 7 cytochrome P450 genes and the hepatocyte marker albumin were used to examine expression levels. Gene expression results were obtained by screening the hepatocyte-like cells against the indicated concentration of each compound and comparing the result against a control treatment. The relative normalized expression, or fold change in gene expression, was automatically calculated using CFX Manager version 3.0. The error bars represent the standard error across 2 biological replicates. After screening, 3 of the compounds, rifampicin, omeprazole, carbamazepine, were selected to further investigate the effect of compound concentration on the expression of a subset of genes. Hepatocyte-like cells, treated with a range of omeprazole concentrations, exhibited a trend of CYP1A1 and CYP1A2 induction, consistent with the screening experiment. CYP3A4 was also induced to a minor degree at 62.5 micromolar. At the highest concentration, 7 of the 8 genes show a drop in expression, including albumin, which normally shows stable expression in hepatocytes. This likely indicates non-specific inhibition of protein expression at 250 micromolar. Cells treated with the same concentration range of carbamazepine show relatively little change in expression until higher concentrations are reached. At 250 micromolar, the expression results agree with those observed in the screening experiment. When the cells were treated with the same concentrations of rifampicin, albumin and CYP2E1 expression steadily decreased, while CYP3A4 expression steadily increased across the concentration range. CYP1A1 expression remained relatively stable until it was induced at 250 micromolar. A narrower range or higher concentrations might be useful to further investigate the effect of rifampicin on CYP1A1 induction. Relative quantification results for the screening experiment were calculated using both the 1-step and 2-step qPCR approaches. Both methods show close agreement, thus demonstrating that using Bio-Rad's 1-step RT-qPCR approach enables rapid generation of high-quality qPCR data. To demonstrate the utility of using the cell lysate approach, we also compared relative quantification results produced using cell lysates and Orum purified total RNA. The data produced using both methods show strong agreement across 15 PrimePCR assays. We hope this provides you with a clear understanding of how the use of one-step RT qPCR kits, in conjunction with cell lysates, can provide comparable performance to a longer two-step workflow that includes RNA purification. Furthermore, this method provides a cost savings and can work with a limited number of cells, as eliminating the purification step prevents loss of template. To learn more about the products used in this project, please visit our website.